Good morning, everyone. Welcome tonight to Western Illinois University's 2024 Morrow Distinguished Lecture. Some of you may not know me. I'm Sean Meager. I'm a professor in biological sciences. Our speaker today on the far side is Brian Allan. But before I introduce him, I want to say a couple of administrative things. A lot of people are zooming in, so all of you please mute. The second thing I want to say something about is the funding for this talk. We have money from the Roger and Jean Morrow Distinguished Lecture Fund, and I actually called up the foundation office and had them do a little research for me. So what I know is this is an endowed fund provided by two faculty members that worked here from 1938 to 1969. One was Jean Morrow. She was one of WIU's first female faculty members, and she worked in biological sciences. Her husband was Roger Morrow, who was the first head of physics, and he did that for 17 years. So what happens with this Morrow lecture is biology and physics switch every year and invite a special speaker from outside. So watch for physics next year, okay? All right. I want to thank other people, especially ones that helped coordinate this event. So Dr. Musser introduced himself. He's the chair. He was critical to getting this going. He's also on the Morrow committee with me, as is Dr. Maggie McPherson is there, and Dr. Vaskar Nepal, who's not here. So that's the committee. I got a lot of logistical help setting this up from April Houston, who is the Biological Sciences Office Administrator, and also from the College of Arts and Sciences Office, Susie Fowler and Don Ebert helped me a lot with planning and budgetary help. Finally, the WIU Marketing and Public Relations Department helped a lot with promotion, like making posters and getting press releases for this event. And I also want to thank Dr. Keith Bachman, Associate Dean from the College of Arts and Sciences, for coming out and supporting us tonight. So thanks, Keith. All right. I think that's all the background. So I still have to introduce the speaker, right? Okay. So tonight's Morrow lecture, our guest is Dr. Brian Allan. He got his bachelor's degree at the University of Michigan and his PhD at Washington University in St. Louis. He currently works at the University of Illinois Urbana-Champaign. He is a professor of entomology, so study of insects, in the School of Integrative Biology. He is also an associate director for academic affairs. Dr. Allan is a broadly trained ecologist. His work focuses mainly on the ecology of infectious diseases, especially ones transmitted by vector organisms like mosquitoes and ticks. He has done work in North America, South America, and Africa. He has written over 60 scientific papers on his ecological work. His work has also been featured in lots of public media outlets like the New York Times and Scientific American. At the University of Illinois, he's also committed to teaching and has received several recognitions for his work on that. So Dr. Allan's talk tonight is the effects of global change on vector-borne disease. Dr. Allan. Thank
you so much. This is a really cool opportunity to come speak to you about a topic I'm passionate about, which are diseases transmitted by insects and other arthropods. It's a recently severe problem globally, and so it's an opportunity for those of you who are students in biology or other fields to think about how we can take our knowledge of biological complexity and apply it to real world problems. And so I'm going to be speaking to you today about a variety of aspects of the way humans are affecting the planet and how that affects our exposure to vector-borne diseases. Normally you save the thank yous for the end, but I'm so grateful I just can't help myself. So first I want to say thank you to Sean Meager for the invitation to speak in this seminar series, and to the rest of the Morrow committee. This is a really kind invitation, and so I'm thrilled to be here. I've had a lovely visit with the Department of Biological Sciences today, so everybody who I met with and who took the time to chat with me, thank you so much. This is a really special seminar series that you have, and so of course credit to Roger and Jean Morrow for having endowed the funds to have this opportunity to have biology and physics take turns hosting speakers from off campus. And then thank you all for coming out on a Thursday night. I know there's lots of good things you could be streaming right now, and so I appreciate you saving those for a little later tonight and giving me an hour of your time. I'll try to make it interesting. No, you're fine. Perfect. Thank you. So I'm a disease ecologist. This is me in the line of work. So this is a region of the world I've worked in for a long time. This is central Kenya, where I've studied ticks and tick-borne diseases since I graduated college. I got really fortunate. I graduated from the University of Michigan. I wanted to take a little bit of time off before going to grad school. And a really amazing opportunity fell in my lap, which is a longtime research mentor of mine offered for me to go run her research program in Kenya for a year. And so after I graduated from undergrad, I moved to Kenya and I spent a year falling in love with East African ecosystems. And so when I finally had the opportunity to return as a professor, it was a really exciting moment for me, kind of full circle, to go back to this place that I had worked for years. In Kenya, I studied ticks, and so these are the tools of the trade. This is a drag cloth on the lower photo here. This is literally a piece of cloth you drag through the vegetation, and ticks that are sitting there in the vegetation waiting for a host to come by will grab onto it. Of course, they don't just wait for the cloth, and so I'm also part of the collection apparatus. And so here I am wearing a tick suit, a white tick suit. That makes it easier to spot the ticks that get on my person. So this is part of the technology that I use in conducting my research. Say what you will about my life choices. This is a field with job security. I didn't know this when I went into it as an undergraduate, but this was the beginning of a period of an explosion in the occurrence of vector-borne disease. So the last 20 years has seen this incredible increase in vector-borne diseases. Here in the US, just during that window of time, human incidence of vector-borne disease has tripled. So by vector-borne disease, I'm referring to diseases transmitted by insects and other arthropods, so things like ticks and mosquitoes. And so we're just increasingly awash in these vector-borne diseases. Here in North America, it's things like Lyme disease and West Nile virus, but there's also things that we historically have thought of as being tropical diseases like dengue virus that are expanding in distribution. This is an article from the New York Times from just a few days ago. Here in the Americas, meaning North, Central, and South America, we're on track to have the worst year of dengue virus in recorded history. So dengue is a mosquito-borne disease that is really exploding. I had some friends who went to the Caribbean over spring break. A bunch of them came back with dengue. It's a very serious virus. It can make people severely ill, can even kill them. We're also discovering new pathogens. So a tick that occurs here in Illinois and other parts of the central US, the Lone Star tick. Recently, multiple new viruses have been discovered that have been associated with this tick, including Heartland virus. And so the big question that a lot of us are trying to answer who work in this field is, why are all these vector-borne diseases increasing in prevalence? So why are we experiencing such a big increase in incidence of vector-borne disease relative to historical incidents, especially considering that we have so much more medical technology than we had before? So as our ability to cure or treat diseases has improved, why are we actually experiencing a greater incidence of vector-borne disease than we did in the past? One reason, and a reason I'll talk about in my lecture tonight, is climate change. And if you want to read a good article about this, this is in Rolling Stone, which is not, I normally associate that with music, but this actually is a lovely article about all the different ways that climate change is impacting the prevalence of infectious diseases, including diseases transmitted by ticks and mosquitoes. And so climate change is part of the story that I'll talk about tonight. But I won't start there. I'll actually end there. And that's somewhat deliberate, because I think we have multiple types of environmental change, multiple types of global change that are affecting the incidence of vector-borne disease. I don't think these things are happening independently. In fact, many of them interact with each other. One is that we're changing the land cover of the Earth's surface. So human activities disrupt natural ecosystems, things like deforestation, changes land cover in ways that can affect transmission of vector-borne disease. So I'll start there. Not only are we in an era of human globalization, but we're also in an era of biological globalization. So because of human activities, many species are gaining a global distribution. And the mosquito vector Aedes aegypti that transmits dengue, among other pathogens, has been transported globally through human commerce. And so because the mosquito that transmits the virus now has a global distribution, the pathogen is able to attain a global distribution as well. So I'll talk next about globalization. And then finally, I will talk about climate change and some of the ways that we're already seeing climate change impacting the distribution of vector-borne disease, as well as what we can expect for the future as human activities further change the climate. So I'll start with talking about land use and landscape change. And my transition slides here feature art by an artist named Jill Pelto, who does work about global change that I really love her artwork. So I'll start by talking about the vector-borne disease that I'm dearest to my heart, which is the ecology of Lyme disease. It's dearest to my heart because my undergraduate research was on the ecology of Lyme disease, and I never looked back. So this was the disease system that made me fall in love with vector-borne disease, as twisted as that sounds. If you're not familiar with it, Lyme disease is caused by a bacterium, a spirochetal bacterium called Borrelia burgdorferi, transmitted by the bite of ticks. Entomologists refer to these as the black-legged tick. You likely may have heard it referred to as the deer tick. Deer tick's a bit of a misnomer. They do feed on deer in the adult life stage, but in juvenile life stages, like the larvae and nymphs you see at the top of that photo there, they prefer to feed on small mammals like the white-footed mouse. And the white-footed mouse is actually the most competent reservoir host for the Lyme pathogen, meaning that a tick that feeds on an infected mouse is very likely to pick up that pathogen and have the potential to transmit it to a human. Deer are not good reservoirs for the Lyme pathogen, but they are important to the tick life cycle. It's a cool disease from a biology perspective. Obviously, it's not cool to have Lyme disease, but it's a cool disease from a biology perspective because it's very ecologically complex. Many different wildlife species are involved in the transmission cycle, in addition to the ones that I show here, in part because the tick will feed on many different hosts. And it's one of the most prevalent vector-borne diseases in North America. It's economically the most impactful. Close to 500,000 people are exposed to Lyme disease in the US every year, not to mention high prevalences of Lyme disease in Canada and in Europe as well. And it's expanding considerably in distribution. We know this in part because Lyme disease is one of a number of reportable diseases, meaning when a physician diagnoses a patient with Lyme disease, they can submit a report to the Centers for Disease Control and Prevention, who tracks the occurrence of Lyme disease cases. The CDC makes these available as interactive maps. And so if you're ever interested in something, for example, for a class project to look at how something biologically has changed over time, Lyme disease is expanding enormously. Historically, there have been two foci of incidence of Lyme disease, one in the Northeast and one in the upper Midwest. But just from 2001 to 2018, each of these blue dots refers to an individual human whose Lyme disease case was reported to the CDC. And you can see the number of blue dots is increasing and the geographic extent of the blue dots is increasing as well. And part of that is because the Lyme disease vector, the black-legged tick, is spreading to new areas. So as the tick spreads to new areas, it's able to infect more people with the Lyme pathogen. Here in Illinois, Lyme disease was not present until quite recently because the black-legged tick was not present until quite recently. The Illinois Department of Public Health maintains a really nice interactive website where you can actually look at the distributions of different tick species and the diseases associated with them throughout the state of Illinois. So again, another source of information for those who are interested. The black-legged tick was only first discovered or detected in Illinois in 1989. It was picked up in Joe Daviess County, right here in the northwest part of the state, closest to the population in Wisconsin, that we now know has been spreading southward over time. It now covers over 2/3 of the state. And with that colonization of this landscape by the tick, it's brought Lyme disease with it, and more people are getting Lyme disease. If you've ever encountered a tick, you likely know that they can't fly and they can't run very far. And so how are these ticks moving such long distances? How are they being dispersed through the landscape? It's not on their own. Something has to be moving them. And it's very likely wildlife and wildlife movements that are responsible for this spread of the tick. But there's quite a few things that humans have done to modify the landscapes in which we live that are likely facilitating the spread of Lyme disease. So we're making it more convenient for wildlife to move through the landscape in such a way that they're dispersing ticks to new areas. One is that during the period of deforestation in the Midwest and the Northeast that took place during the 1800s and 1900s, we eliminated a lot of the habitats, not just for ticks, but for wildlife. And so as those forests have grown back, so have the habitats returned in which these ticks and their wildlife hosts can live. There's also an increasing pattern where people want to live in more natural places. So patterns of residential development in areas that are somewhat wild create the opportunities for humans to come into contact with ticks and for wildlife to live in close proximity to humans. Just driving back from dinner tonight, I saw some deer on your campus, which is always lovely to have a wildlife encounter. People who see how prevalent deer are now might not appreciate that deer were nearly hunted to extinction in the early 20th century in the US. And so deer were almost wiped out. They were extirpated from much of the continental US. And with the recovery of deer populations due to the imposition of hunting regulations, with an increase in deer and other wildlife hosts, we've seen an increase in ticks. And then finally, a slightly more complicated story that I'll tell you relates to forest fragmentation, the process by which people carve up the landscape and leave little patches of forest behind. And those little patches of forest turn out to be absolutely ideal habitat for ticks for a reason I'll explain to you next. So here's an image, not from Illinois, because there's ocean in the background, of a fragmented landscape. So it gives you an idea of what it looks like when human development leaves behind a patchwork of forest. Most species of wildlife cannot persist in those small forest fragments. There's not enough habitat for them to complete their life cycles, but there's a few animals that really benefit in that fragmented context. One is the white-footed mouse, which I mentioned already is a key reservoir host for the Lyme pathogen. White-footed mice, as it turns out, thrive in small forest fragments. We know this from mammalogy research here in the Midwest, where people studied the relationship between the density of mice, which is depicted on the y-axis in this graph, and the area of forest fragments, which is depicted on the x-axis. What you can see is that small forest fragments have much higher densities of white-footed mice than do larger forest fragments. And the reason we think that's the case is because most other animals can't live in those forest fragments. So the foxes or the owls or the other animals that would control mouse abundance aren't able to survive in those really small patches. And so mice experience explosive population dynamics in those small patches. With a lot of mice, you have a key host feeding ticks. So these are larval life stage ticks embedded in the ear of a white-footed mouse. Those larvae that feed on a white-footed mouse will have a high probability of picking up the Lyme pathogen, molting into a nymphal life stage tick that can transmit Lyme disease to humans. And indeed, when you look at the relationship between forest fragment size, again, on the x-axis, but now the density of black-legged ticks infected with Lyme disease on the y-axis, we see a very similar pattern. These small forest fragments have very high densities of infected ticks, presumably because many of the ticks in those small forest fragments are feeding on this ideal host for the Lyme pathogen. So forest fragmentation is another form of landscape change that seems to be increasing the prevalence of Lyme disease. And with residential development and other forms of land use, we're seeing a lot more fragmentation of forested habitats. So a lot more potential for people to get Lyme disease, to contract Lyme disease in these fragmented landscapes. I went into depth about Lyme disease because it's my favorite, but there are plenty of other examples of where landscape change has impacted vector-borne disease risk. A very similar story is playing out right now in the Amazon forest in South America, where deforestation is rampant. And these logging activities, it turns out, are causing a reemergence of malaria in South America. So malaria is a mosquito-borne disease. There's no wildlife host involved. It's transmitted from human to human by the bite of an infected mosquito. But this deforestation that's taking place in the Amazon is creating a bunch of mosquito habitat. In particular, when these areas are logged, they leave behind small puddles. Those puddles receive direct sunlight, and they're the perfect habitat for the South American malaria vector mosquito, Anopheles darlingi, to lay its eggs. Its larvae develop in these puddles. The direct sunlight results in a lot of bacterial growth, which is food for those larvae. And so with that logging activity, you're creating ideal habitat for Anopheles darlingi. And as a result, malaria has spiked in areas in the Amazon that are experiencing deforestation. We know this in part because of the work of entomologists studying the rate at which they were bitten by malaria mosquitoes. And that is exactly what it sounds like. They literally sit there and count the number of times they get bitten by malaria mosquitoes. But if I haven't sold you on this career yet, I will by the end of this talk. And so comparing across forest cover along the x-axis there, these are sites where forest cover is mostly depleted, so only 0 to 20% remaining, compared to sites where forest is largely intact. You can see that they're getting bitten by Anopheles darlingi at a much higher rate in areas that have been deforested. And so that creates all these opportunities for malaria transmission among people. Another concurrent process that's connected to land use and landscape change is globalization. The fact that not only are humans an increasingly globalized species, but we're making other species that live on the planet Earth global in distribution as well. This also ties in with a slightly more complex concept called biotic homogenization, I'll explain it in more detail through an example concerning West Nile virus, but it's the basic idea that we're making wildlife communities more similar to each other, that by moving species around the planet, we're creating communities of wildlife that all look the same. A really good way to visualize this is if you went to a forest in Illinois and a forest in California, you'd see very different bird species in those two forests. But if you go to a city in Illinois and a city in California, you'll see a lot of the same bird species. You'll see robins, you'll see sparrows. These are things that can persist in a human-dominated landscape. That's the process of biotic homogenization. We're making all of nature more similar through our activities. Perhaps the biggest problem in globalization of vector-borne disease has been that through human commerce, we have transported vector species all around the planet. So species that used to have more limited distributions have been transported to new places. Those are places they probably wouldn't have easily arrived on their own, but they're suitable habitats. Two of the most problematic along these lines are two species of mosquitoes, the yellow fever mosquito, Aedes aegypti, and the Asian tiger mosquito, Aedes albopictus. And they're collectively responsible for transmitting a host of problematic viruses. Dengue virus arguably being the worst of all these in terms of human incidence and mortality, but also some diseases you may have heard of like Zika virus, chikungunya virus, and even the old scourge yellow fever. The reason why it's been so easy for humans to move these mosquitoes around the planet is because they have desiccation-resistant eggs. What that means is that their eggs can survive drying out. And so female mosquitoes will lay their eggs in a habitat that's not yet wet, so that it hasn't become inundated with water. But then when rainfall fills up that habitat, their eggs hatch. And so because of that, their eggs can be transported all around the earth. And then all that has to happen is rain has to fall on that habitat and those eggs can hatch. And it turns out these two species of mosquitoes love laying their eggs in tires. Tires are like their preferred habitat for the development of their larvae. And so the global tire trade has moved these desiccation-resistant eggs all over the planet. As a result, Aedes albopictus, which is more temperate in distribution and has actually invaded much of eastern North America in the last 15 years, and Aedes aegypti, which is more tropical in distribution, now cover a significant percentage of the Earth's land surface. What that means is if somebody with one of these viruses gets on an airplane and flies somewhere else and gets off that airplane, they can start a new pandemic or a new epidemic in that place by bringing that virus to a place where the mosquito is already present, but the virus has not yet been introduced. We saw a striking and somewhat terrifying example of this with the invasion of Zika virus in South America that began in 2015. So in 2015, a new disease was discovered in Brazil. And it was discovered because of an outbreak of a very unfortunate and incurable condition, which is infants born with microcephaly, unusually small and underdeveloped heads and brains. It's a lifelong condition, it's incurable. There are other things that can cause microcephaly, but it typically occurs very rarely. And in Brazil, in March 2015, physicians noticed an incredible spike in microcephaly and realized it was likely that some new disease had been introduced. Up till this point, Zika virus was really off most scientists' radars. I studied ticks and mosquitoes, and I wasn't particularly concerned about Zika virus. I did not realize its potential to spread to new places and cause outbreaks. But of course, it's primarily transmitted by Aedes aegypti, and because of the global distribution of that mosquito species, Zika virus is now all over the planet, where it can cause large outbreaks in immunologically naive populations. Zika virus is, I think, the classic example of how we may need to revisit diseases that in the past were not considered to be major or important diseases. It was first discovered in 1947, almost by accident, as part of a medical study using rhesus monkeys. A few other cases or outbreaks were reported in South Asia over time. And then it started hopping its way across the Pacific. And so in 2007, there was a sizable outbreak in Micronesia, still not really big enough to catch attention of most biologists or medical experts. But then a big outbreak in French Polynesia in 2013 was the first time that people realized maybe Zika wasn't that mild a virus. It was the first time that there were reports of microcephaly, other more severe cases. People had assumed Zika was mild, but I think that assumption was false, in part because Zika was occurring in places where people had been exposed to it before. So it didn't cause a severe disease in the places where it was established compared to the places where it arrived later. And then somehow it jumped to Brazil, and in 2015 was detected, and then rapidly spread throughout the Americas, causing a very large outbreak. What's amazing is we now have the scientific tools to reconstruct this introduction event. So we can actually figure out how was Zika introduced and where did it come from. Part of this is through an evolutionary tool called phylogenetic analysis. That's where we study the genetic relatedness of different strains of the virus to try to figure out the current strain in Brazil, from what strain did it evolve? And scientists were able to confirm that it evolved from the French Polynesia strain, and that the timing of when the outbreak in Brazil began coincided very well with the peak in the outbreak in French Polynesia. These scientists very creatively were also able to associate it with human travel associated with sporting events. So Brazil hosts a lot of big sporting events, and the timing of the introduction happened to coincide very well with a major soccer tournament that was taking place in Brazil at the time. And so we think people traveling from French Polynesia to attend this soccer tournament brought Zika with them. One more piece of evidence in support of this is that these scientists actually looked at airline travel and compared the flow of humans from French Polynesia to Brazil at that time, and indeed found that around when there was a spike in airline travel is when this outbreak began. That's all pretty incredible. The one other thing that came from this that strikes me, though, is if you look at that point number 2, a single introduction event, that sounds really benign, right? What the genetic data suggests is it was one human who got on a plane in French Polynesia, infected with Zika virus. Zika virus is often asymptomatic. The person likely didn't even know that they were sick. Got off of a plane in Brazil, got bitten by an Aedes aegypti mosquito, and started a massive outbreak that spread throughout the Americas. We can figure that out based on the genetic diversity of the early strains of the Zika virus outbreak in Brazil. They were all incredibly similar, suggesting just one introduction, just one person traveling from French Polynesia to Brazil started one of the biggest outbreaks of the 21st century. Incredible what we can do, but also daunting in terms of what it means from a prevention and control standpoint. Another similar story of a mosquito-borne disease and globalization is West Nile virus. So West Nile virus is now the most prevalent vector-borne disease in North America, but 25 years ago, it wasn't even here. It was detected in New York City in 1999 in the proximity of the Bronx Zoo, where some veterinarians working at the Bronx Zoo noticed a massive die-off of birds. That birds started sort of dropping from the sky. And there was also an outbreak actually at the Bronx Zoo in their bird population. And that's when they tested some of these dead birds and discovered a virus that had never been reported in North America before, which is this mosquito-borne virus called West Nile virus. Well, diseases like Zika and malaria are transmitted from human to mosquito to human. West Nile virus is more like Lyme disease, where it has a wildlife transmission cycle. So West Nile virus naturally is transmitted among birds by mosquitoes in the genus Culex. And so an uninfected mosquito bites an infected bird, picks up the virus, that mosquito becomes infected and can continue to spread the disease. Humans are actually an incidental host. West Nile virus isn't very well adapted to us, which might be one reason why it makes us quite sick, is because we don't have any significant evolutionary history with this virus. But what was amazing is from 1999 to just 2006, West Nile virus swept across North America. We're not entirely sure how, but it's likely through movements of migratory birds dispersing the virus from one side of the continent to the other. And so in a 6-7 year window, it went from being non-existent in the US to becoming the most prevalent vector-borne disease in North America. It's an urban disease because, one, the habitat associations of the mosquitoes. So we often think of mosquito-borne diseases as something we associate with wild places. But in fact, if you're going to get West Nile virus, you're very likely to get it in a city. One reason for that is that the mosquitoes that transmit West Nile virus take advantage of a human-made habitat, stormwater infrastructure. So if you've ever walked down the street, you've likely walked past one of these storm drains. You probably don't think about it past the point of that's where water goes when it rains. But there's this device underneath that grate. It's a stormwater catch basin, and it's designed to capture rainwater during a storm event, so all that water doesn't flood the streets or end up in our basements. Unfortunately, that is an ideal habitat for urban mosquitoes to lay their eggs and for their larvae to develop. These catch basins fill up with debris, leaves, and other trash. And as microbial growth develops on those leaves, that's food for mosquito larvae. And so most Culex mosquitoes in urban environments are developing in these stormwater catch basins along the roadside. So that's one reason why it's an urban disease. The other relates to that example I gave about biotic homogenization, the idea that we're making wildlife communities more similar. It turns out that for West Nile virus, some bird species are really good reservoir hosts, so they transmit the virus to mosquitoes at a high rate, while other bird species are very incompetent reservoirs. They get infected with the virus, but they don't pass it on to mosquitoes. The bird species that turn out to be really good at transmitting West Nile virus are also urban bird species. And so by homogenizing these communities, by having cities where we attract just the same kind of community of birds over and over again, we're creating this perfect storm where both the mosquito that transmits the virus and the hosts that carry the virus are both associated with urban habitats. And so that homogenization of wildlife communities is the other factor that makes this an urban disease. If you look at the data on human cases of West Nile virus, they group really strongly with urban areas. So this is a map, again, provided by the CDC. These are West Nile virus cases in 2023. And where you see big clusters of Lyme disease, or sorry, West Nile virus, too many vector-borne diseases, are places like Chicago, Peoria, Detroit, New York City, New Orleans, urban habitats where we've created the same type of wildlife community over and over again through the ways that we've modified the environment. And so that's where a lot of cases of West Nile virus are occurring, especially in the eastern part of the US. And then last but not least is climate change. So I've intentionally left climate change for the end, in part because this is one of several things that's impacting the distribution of vector-borne disease. And when you look at popular media articles, often this increase in vector-borne disease is attributed to climate change. I believe climate change is one of several factors that's contributing to vector-borne disease emergence. And so I'm not discounting the important role that climate change is playing, but the evidence that climate change is the main driver of vector-borne disease emergence is still limited. I will give some key examples, though, because we do have some areas of the world where we think climate change is really driving emergence of new vector-borne pathogens or old vector-borne pathogens as well. Malaria is fascinating because malaria is arguably the oldest disease of humans and the deadliest disease of humans. When you study the evolutionary history of malaria, there's a sister species of malaria that occurs in non-human great apes, and it diverged from the human strains of malaria 4.9 million years ago, right around when we believe humans and chimpanzees diverged in their evolutionary history. So malaria as a disease has been with us for as long as we've been a species, and which is pretty incredible. Also, because of the high mortality rate of malaria, especially in children, we think it's been the deadliest disease in human history, but it's killed more people than any other disease ever has. Globally, we've had enormous success with reducing the incidence of malaria through various vector control programs, drugs that treat the disease. And so in this nice set of maps that you see in the journal Nature from an article published in 2010, this is what the distribution of malaria was globally about 100 years ago, and this is what it was 100 years later. And so it may surprise you to learn that malaria occurred right here in the central US, especially the Mississippi Valley region. People got malaria right here in central Illinois 100 years ago. But through 100 years of medical interventions and technologies as simple as window screens, we've really cut back on the prevalence of malaria. And now the most severe prevalence of malaria is limited mostly to sub-Saharan Africa. What this last figure shows is a hot-cold map or a heat map of where malaria has increased and where malaria has decreased. The blue refers to areas where it's decreased, the red areas where it's increased. As you can see, globally, it's been a real success story. We've really limited the prevalence of malaria in quite a few places. But there are some spots where you see some red, places where malaria has increased, like Nepal, East Africa, and Colombia. And what these areas all have in common is that they're high altitude locations, places where historically they were too high up for malaria mosquitoes to complete their life cycles. But with global warming, we're seeing a lot of warming at high altitudes, and it's allowed malaria mosquitoes to move up in distribution. These are some data from East Africa, looking at highland regions in places like Ethiopia, Kenya, other parts of East Africa, and the percent increase in malaria in these locations as a result of global warming. Some of these are gigantic increases, in part because these were areas where there was no malaria. The highlands of Ethiopia were largely malaria-free pre-global warming, and with climate change, we're seeing a huge jump in malaria cases. And this is not trivial in terms of the number of people who are being exposed to malaria. Ethiopia, I think, is the second most populous country in the African continent, and most people in Ethiopia live at high altitude, which anthropologists believe is actually the historical signature of malaria, that people settle at high altitude to avoid malaria. And so as malaria has moved up in altitude, it's affecting many more people who historically lived in a place that was malaria-free. Among my contributions to vector-borne disease data, I think I'm a data point in this graph. I got malaria in Kenya in 2000, and it was at one of these high altitude locations that historically had been malaria-free. I wasn't taking any anti-malarial drugs because I was told you don't have to worry about malaria. We're above 6,000 feet. And I got malaria like that. And so I believe I may be a data point in this graph, which is a nice accomplishment. Other diseases for which we're seeing signatures of climate change include wildlife diseases as well. This is a disease many people haven't heard of, but it's actually quite prevalent here in Illinois. It's called epizootic hemorrhagic disease, or EHD, and it's transmitted by culicoides midges, also sometimes called no-see-ums. No-see-ums, for most of us, are just annoying. They bite us in the summertime. It doesn't feel great, but they don't transmit many diseases. But for deer, they transmit an incredibly deadly disease, EHD, which in deer populations that haven't previously been exposed to it causes over 90% mortality. So this is like the Ebola of deer, and it's transmitted by midges. Researchers tracking EHD outbreaks for decades are showing significant northward expansion of EHD over time. So in the 1980s, the northern limit of EHD was somewhere around the border between Illinois and Wisconsin. And you can see over time that is tracking northward. Biologists believe that's a signal of climate change, that we're seeing warming at higher latitudes, and that's allowing vectors like these culicoides midges and this virus they transmit to move northward in distribution, bring their diseases with them. And these are deer populations that have never experienced EHD before, so they're highly vulnerable to it. The deer become very thirsty before they die, and so they often die by water. And I've seen dead deer here in Illinois that I suspect to be EHD cases, because you see them in the summertime along riverways and other waterways. The vector-borne or the tick-borne disease, Lyme disease, is likely another example of where we're seeing a change in incidence as a result of earlier springs. As I mentioned, Lyme disease not only occurs here in the US, it's prevalent in Canada, and we think the expansion in Canada is climate change related. And in Europe, they're seeing northward expansion as well, especially at higher latitudes. So in Norway, Lyme disease historically has been most prevalent in the southern part of the country, but it's moving northward. They're seeing an increase in Lyme disease cases over time. And the timing of Lyme disease is getting earlier and earlier in the year. So the public health agencies in Norway track not only the number of cases of Lyme disease, but when they occur. And the peak timing of Lyme disease cases is getting earlier and earlier in the year. And this corresponds with other signals of climate change, like earlier greening. So earlier springs seem to be associated with the tick life cycle starting earlier in the year and getting Lyme disease cases started earlier in the year. When we look at projections of future vector-borne disease distribution, it's discouraging. And so one way that scientists do this is to look at the current distribution of an organism like the mosquitoes that transmit these viruses. This is Aedes aegypti again at the top, Aedes albopictus at the bottom. We look at the current climate they're associated with. We call that ecological or environmental niche modeling. So we make a map of where they currently occur. And then we use climate change projections generated by climatologists to ask where will those climates be in the future. So those regions that are currently possible for them to live in now, where do we expect those to be by, say, mid-century? Unfortunately, by mid-century, Aedes aegypti may get up into Illinois. And so we could imagine there could be transmission of viruses by this mosquito in places like where we live now. And Aedes albopictus could actually spread well up into Canada. And so we could see here in North America a greater northward spread of these mosquito-borne diseases as a result of climate change facilitating the expansion of their ranges. Interestingly, some parts of the tropics where these mosquitoes are currently most problematic may actually become too hot for those mosquitoes to live. And so we may actually see a poleward shift in some of these vector-borne diseases that we think of being tropical diseases, but they're of course tropical in a previous or historical climate and not a future climate where they might look quite different. So thinking about all the different ways vector-borne diseases are expanding in distribution can get a little depressing, and it makes us question, what does the future hold for human health? And so I want to sort of wrap up by talking about what I perceive as being some of the solutions to these problems. And some of these are encouraging solutions because we know what we call win-win solutions. Solutions that not only benefit human health, but maybe have some other environmental benefit as well. One simple method for tick control is actually one I'm quite fond of. It's called the four-poster apparatus. It's a feeding station for deer. And in the process of coming in to feed, the deer receive a topical pesticide that kills ticks. So this column is filled with corn. There's a little tray here where the deer come up to feed. These are just paint rollers that a scientist has to come out periodically and top up with pesticide. And then when deer come to feed in this basin, they get the pesticide all over their face and their neck, and then through grooming, they spread it over the rest of their body. And so these 4 posters are a very simple but effective technology for killing ticks directly on a wildlife host, like a white-tailed deer, which serves as an important host for the life cycle of a number of tick species. We're also finding that some of the other environmental problems that we're trying to solve also carry some benefits in reducing vector-borne disease risk. If you're not familiar with bush honeysuckle, just go outside right now, look at the woods. If you see something that's green, that's pretty likely to be bush honeysuckle. Just on my drive today from Champaign-Urbana, I saw it everywhere. Bush honeysuckle is a very noxious invasive shrub, and so there's a lot of efforts to try to control this plant. Here's my colleague Umberto Dutra doing a bush honeysuckle removal. And as part of this bush honeysuckle removal experiment that we conducted in St. Louis, Missouri, we discovered that when you remove bush honeysuckle, the risk of tick-borne disease drops dramatically. So in areas that are invaded by bush honeysuckle, there's high densities of ticks infected with pathogens. And when you remove the bush honeysuckle, the risk of tick-borne disease drops a lot. We think this is happening because wildlife hosts like white-tailed deer congregate in honeysuckle-invaded areas. And so here's an example of a win-win where removing an invasive plant species can also come with the benefit of reducing human risk of exposure to a tick-borne disease. There are some possible win-wins for mosquito control as well. And my favorite example of this is green stormwater infrastructure. And so as I mentioned at the beginning of my talk, these urban mosquitoes that transmit West Nile virus have adapted to completing their life cycle in the stormwater management tools we use for managing stormwater, like catch basins. For reasons completely unrelated to mosquitoes, though, there's a movement to try to replace that type of conventional stormwater infrastructure with what's called green stormwater infrastructure. And green stormwater infrastructure are any technology that mimics the natural hydrology. And so, for example, this permeable pavement in this parking lot, instead of capturing all this rainwater in a catch basin, the water can just flow right through this pavement into the soil. Likewise, rain gardens, which I suspect you likely have right here on your campus, are another example of green stormwater infrastructure, a natural habitat where you allow water to pond during a rain event, but then it filters down into the soil. Stormwater engineers are replacing conventional stormwater infrastructure with green stormwater infrastructure because it improves water quality. So it makes water cleaner, it reduces the amount of pollutants that go into surface waters. But as we're discovering, these types of technologies can also control mosquitoes by removing mosquito habitats. We discovered this through a study we conducted in Aurora, Illinois, where there happened to be a large-scale replacement of conventional stormwater infrastructure with green infrastructure. And it's a really simple switch. They're replacing conventional catch basins, which are these habitats that are perfect for larval Culex mosquitoes to develop, with infiltration catch basins, ones that have a permeable bottom. And so they work the same way. The rainwater goes into the catch basin, but then instead of sitting there for a week while a mosquito can complete larval development, the water just filters out through the bottom into the soil. And so my colleagues and I took advantage of a green infrastructure project in Aurora where conventional catch basins were being replaced with infiltration catch basins. We identified another area that was not being replaced to use as a control. And we studied what happened to mosquitoes when green stormwater infrastructure was implemented. What we found before the green infrastructure was implemented was that the catch basins that would become green infrastructure, the green ones in this graph, held just as much water as the conventional ones. But when they were replaced with infiltration catch basins, the amount of water they held dropped considerably. So infiltration catch basins work. They don't hold as much water as conventional catch basins, exactly as designed. When we looked at the abundance of mosquitoes in these habitats, what we found is that the ones that the engineers had identified to replace were actually some of the most productive habitats for mosquitoes. They were in green here, producing the most mosquitoes. After they were replaced with infiltration catch basins, we almost never saw mosquitoes in that green infrastructure because there was almost never any water in that green infrastructure. So it just eliminated the mosquito habitat, still managed the stormwater, got rid of the mosquitoes. So another example of a win-win solution. So I'll wrap up by saying that we are living in challenging times when it comes to vector-borne diseases. Through human globalization, landscape change, and climate change, we are seeing an increase in vector-borne disease, which is still somewhat surprising even to me, considering the strength of the technologies that scientists have. But if we really understand why these vector-borne diseases are expanding, what's causing them to increase in incidence, I think we can come up with solutions, technology solutions, biology solutions that can allow us to at least mitigate the worst effects of vector-borne disease And so I think for anybody who's interested in going into this field, making a career out of this, there's really a lot we can still do to try to manage the burden of vector-borne disease on humanity. I'll say thank you again, Western Illinois. It's been a really fun week.
Thank you, Brian. So there's time for questions, if anyone has them for Dr. Allan, if you'll take them.
Yeah, any questions? Go for it.
Great talk. Thank you. Thank you very much. So I have a bunch of questions, but let's start with first one. There was a graph where you show the detections of Lyme disease. You mentioned that we're on humans, right? I have a dog and went to the vet and got the blood test and got Lyme disease positive. So the question is, do you know if the data from pets is being recorded and used in this type of studies?
Yeah, I really appreciate the question. So the question is, are we collecting any veterinary data on diseases like Lyme disease? Many of our pets can get the same vector-borne diseases we can, and so is that a resource? I was doing a study on the geographic expansion of Lyme disease, and I tried to use the veterinary data, and it wasn't as carefully organized as the human case data. So there is a private company that organizes the Lyme disease case data for dogs. It is more presence absence than more detailed data like the number of cases, the number of patients. So unfortunately, I think we're missing out an enormous source of information by not having a reporting system for our pets akin to the reporting systems we have for humans. West Nile virus makes horses incredibly sick. Lyme disease makes dogs sick. These are sentinels, animals that can get the same diseases that we can, and we could use those data to better understand these vector-borne diseases, but there hasn't been the same push to organize those data into a national database the way there has been with the human data. Even the human data, we think, is only about 10% of the cases. And so I mentioned at the beginning of my talk about 500,000 cases of Lyme disease per year. Only about 30 to 40,000 are reported to the CDC in a typical year. And the reason we're able to estimate that there are 500,000 is based on insurance data, the number of humans who claim disability as a result of having had Lyme disease. So even the human data is actually just the tip of the iceberg. Thank you. That's a great question. Yeah, at the back.
I work with equines, and I do show some concern with the West Nile especially, but working in Texas and in Florida with international competition, there has been notice that there may be potential that there's a mutation going on with West Nile virus and showing resistance to vaccine. Is there any data that there may be a transmission that may happen where the mutation may come to humans or make it zoonotic, or is there any sign of mutation that is occurring within these vector-borne viruses?
Oh, that's a fascinating question. I'm actually not aware of the genetic data indicating that West Nile virus is changing to that extent. That is concerning because, yeah, we are able to develop vaccines for non-human animals much more quickly than for humans. And so pre-vaccine, I think the horse mortality from West Nile virus was like 1 out of 3 animals died. But they were able to get a vaccine to market very quickly, and it was very protective. So I think that would be bad news if West Nile virus is able to evade that vaccine protection. I guess the good news is because we don't have the same regulatory systems for developing animal vaccines that we have for human vaccines, I would be hopeful that a new West Nile virus vaccine could be developed if that were the case. Yeah, thank you. Yeah.
You mentioned when in the 4-post thing with the deer, you mentioned that they coat the deer with insecticides. I know typically insecticides are associated with a lot of negative impacts for like the environment and creatures and stuff like that.
Yep.
So with the insecticide, has there been any negative side effects of that in any area?
Yeah, fantastic question. So the question is, some of these pesticides that we use, including that we might use on wildlife, might there be any negative impacts? For the 4-poster technology, the pesticide that's being used is called permethrin, and it's actually approved for use on humans as well. You're not supposed to spray it directly on your skin, but on your clothing. So it is our best tick repellent that's out there. It is definitely something that we need to be really aware of, that when we introduce these types of chemicals into the environment, there could be negative consequences. The example I'm most familiar with is that previous generations of pesticides that were much less target specific, like organophosphates, really caused a lot of harm to the environment. And so in another part of the world where I work in Kenya, they were using organophosphates on cattle, and it was actually causing declines in other livestock species, especially birds that eat ticks. So birds that were eating ticks off of cattle were actually getting poisoned by those organophosphates. And so I think the upside is we've gotten more advanced chemicals for these types of tasks that are more specific to particular targets. But I totally agree with your point that we shouldn't take it lightly, that there could be some negative consequences of using these chemicals.
Dr. Allan, might be a question.
Yeah, for sure. Let's check the chat. Okay, so Joy says, now we know that it is a vector-borne disease. It is a great problem for public health entomology, but what practical things can we start doing as individuals to help solve the problem? And are there organizations we can work with for further research or even on field experience, especially for like an internship? Yeah, great question. So there are things you can do to control vectors and vector-borne diseases around your own home. And in fact, for mosquito control in urban and suburban environments, that is arguably the most important thing, is to get homeowners to buy in and help contribute to the control of mosquitoes, because it's really a house by house kind of thing. And public health officials in this nation and in many others can't necessarily just walk into somebody's backyard and treat the mosquito habitat in the backyard. So we really need buy-in from homeowners. Luckily, there are lots of things you can do. If you have small aquatic habitats on your property, like bird baths or rain barrels that you use to capture water and use it to water your garden. There are mosquito dunks that you can buy off of Amazon. They look like a little donut, and you throw it into that water basin and it dissolves slowly over a month. What that mosquito dunk contains is one of the most effective insecticides ever developed. It's called BTI. And BTI is a toxin produced by a bacterium that's specific to the Diptera, so it's specific to flies. So it really only kills mosquitoes, midges. You could literally take a cup of that water and drink it yourself, and it would have no harmful effects. And so it's like the least harmful insecticide. It works beautifully, and it's not prone towards developing resistance. And so we don't see any sign that the mosquitoes that we use to, that we control with BTI are developing resistance against it. So anybody with a bird bath or any other kind of water habitat on your property, I always say, go on Amazon, buy a 6-pack of mosquito dunks. That'll get you through a whole summer. There are things you can try to do to control ticks on your properties as well. And so for a lot of us, you know, I think the vision is that when we go out into nature is what we get exposed to these vector-borne diseases. But work on Lyme disease in the Northeast says most people are getting Lyme disease in their own backyard. They're going out to garden, they're going out to work on the edge of their property, and that's where they're picking up ticks. So also the personal preventative measures you can take, like wearing repellents or checking yourself for ticks after you've been outdoors, are the best strategies. As for organizations, yeah, there are quite a number. Society for Vector Ecology is very active in the US. They host an annual conference. Here in Illinois, we have a Illinois Mosquito and Vector Control Association. So we actually have a scientific organization here in the state of Illinois that is devoted to tick and mosquito control. And there are lots of opportunities for students to get involved with these types of organizations and this type of research. Questions?
I have another one.
Yeah.
That's one that's asking. So there was a really interesting graph that you show. I think it was the distribution of mosquitoes on the planet.
Yep.
Right? And it was curious to see that in some cases, like in India, it really just looked like if it was just completely covering the country. So and then on the other side, there was nothing, right? So those cases, is it a real edge there of population, or is lack of data? And if it is lack of data, this kind of indicates the issue of politics, right? How it gets into all this, so we can collect the data needed to study, because those places may be in even worse conditions if we don't have the data.
Yeah, that's very insightful. And so, yeah, the white areas on those maps might be white because the mosquito's not there, and they might be white because nobody's looked for it. And in fact, what you were looking at was a probability distribution of the occurrence of the mosquito, because we really only sample in specific places, right? And then you try to turn that into a continuous map of the distribution of the mosquitoes. And so the very red areas were the places where there was a very high probability the mosquito was present. This might be because there are human dengue cases reported from that location, or because an entomologist like myself actually went there and captured mosquitoes. In some cases, it might be geographic barriers like the Himalayas. And so there might be things that these mosquitoes really can't easily disperse across, and it would really require human facilitated dispersal. But we're often finding it's just as much the other issue that people just haven't looked for it yet. And that's why we don't know if it's there or not. And so that's a best estimate of the current distribution of those mosquitoes, just based on the limited data we have.
You had two graphs showing the spread of Lyme disease in the continental United States from the early 2000s to 2018.
Mhm.
In the version from 2018, it had excluded the entire state of Massachusetts, and I'm curious as to if that was like a data fluke or if that they happened to somehow maybe eradicate it or something.
I wish it was the latter, but it's the former. So each state collects the Lyme case data and reports it to the CDC. And in that year, Massachusetts did not report. I also showed 2018 instead of a more recent year because COVID and the COVID pandemic really messed up Lyme disease reporting. And so the data from 2019 after are currently just a mess. And we're still waiting for the CDC to clean up those data. And we use those data. I've used those data as part of analyses. And so I think it's appropriate that the CDC is holding back those data until they ensure that they're correct, given that they know there are researchers who are actively using them. But yeah, you're exactly right. That was a keen eye. That's a data fluke. One more? Okay, at the back.
To argue from I guess a different side
Sure.
of the question. Or if say a state or a city were to bring sanctions or a new architectural plan to admit these I guess to help prevent the mosquitoes and the vector-borne from an economic standpoint. I don't know if you have an idea, but how much from if we were to implement this, how much would it cost a city or a state or even on a national level, especially since many different green projects are going on, especially in automotive and wind energy. Would this be a $1 million, $1 billion, or $1 trillion kind of mark, or would this be like at the end of the day, this price tag will be better off instead of a future price tag of the latter?
Yeah, that's a great question. I take your meaning exactly. I couldn't quote a number for what it would cost to do this. What I would feel fairly confident projecting is that it would pay off. And one reason I think that is because in a lot of places where it's happening, the stormwater infrastructure is failing anyway. So it's a lot of places like the Midwest where the stormwater infrastructure is 100 years old. It's falling apart and it has to be replaced. And so if you're tearing up a 100-year-old catch basin, in my opinion, you might as well replace it with an infiltration catch basin instead of a conventional catch basin. And that's exactly what's happening in the city of Aurora. Those were all stormwater infrastructure that were in decline. And so they had to be replaced. The city of Aurora was also able to get a grant from the federal government to pay for it if they did green infrastructure instead of conventional infrastructure. So it saved the taxpayers of Aurora money having the green infrastructure put in. The other things I would point out is that the costs of vector-borne diseases are incredibly high. Lost productivity, lost wages. People don't necessarily die from these diseases, but when they're sick, they can't go to work. Lyme disease is a classic example of this. Lyme disease is a chronic debilitating illness. And so some people with long-term Lyme disease are never able to return to the workforce. And so the lost productivity is another financial aspect of this that I think I would take into consideration. So I'd feel pretty comfortable saying I think green infrastructure would pay for itself, given that it's all projects that likely need to happen anyway.
So I think we need to end there, so let's thank Dr. Allan again.
And before you pack up, I just want to say, there's a reception for Dr. Allan right now, like around the corner in 121. So if you want to talk to Dr. Allan about anything or each other, or mainly get free food, it's there and it's for you. Okay, so I'm gonna run there and turn on the lights so you know where it is. Thank you for coming, everyone.
Yeah. It's not like it's fine. I was basically. Haven't worked out as much. What let's show this. What? Basically, the president really is been.
Hopefully it's talk.
Yeah. Still got. But it's again,